Today, we're jumping into a book that's, well, it's pretty mind-blowing. It's Walter Isaacson's The Codebreaker, and we're going to unpack the story of CRISPR, a gene editing tool that's on the verge of changing, well, pretty much everything we thought we knew about life, disease, and what it even means to be human. So let me ask you something. Think about your family, your health history. You know those things that seem to run in the family, like heart disease or diabetes, popping up generation after generation? Have you ever just thought of it as, I don't know, bad luck? Just a roll of the genetic dice? But what if it isn't a game of chance at all? What if it's actually just a spelling mistake? A tiny little typo in this huge instruction manual that is your life. This book that decides your eye color, how tall you are, and yeah, whether you're more likely to get certain illnesses. Okay, so before we can even begin to talk about fixing that typo, we've got to understand the language this incredible book of life is written in. Let's get into it. That book, that's your DNA. It's basically nature's software. It's this beautiful, elegant code that has all the instructions needed to build and run you. And it's written with only 4 chemical letters. Every single cell in your body has its own copy of this massive manual. And when I say massive, I am not kidding. We're talking over 3 billion letters long. 3 billion! Now, with a text that enormous, you can start to see how just one single typo, one little letter in the wrong place, could lead to a really devastating disease like sickle cell anemia. Just one mistake and a person's entire life is changed forever. You know, for almost all of human history, all science could do was read this book. That's it. We could find the typos, we could figure out what they did, but we were totally powerless to fix them. We just had to accept our genetic fate. But that has all changed. We are not just readers anymore. We've become the editors. So how on earth did this happen? How did we get this almost godlike power? Well, the story doesn't start where you might think. Not with a big quest to cure human disease, but with a really simple question about one of the simplest life forms on the planet. You know, every huge discovery has a human story at its core, and this one is no different. It's all about 2 amazing scientists from totally different parts of the world, Jennifer Doudna in America and Emmanuel Charpentier over in France. And they were both completely fascinated by this really basic puzzle. How in the world do bacteria defend themselves when viruses attack? The answer, it turns out, would change, well, everything. What they found was just this incredibly clever system that nature had already figured out. So picture a bacterium as a little soldier. A virus attacks, right? If the bacterium survives, it does something amazing. It actually cuts out a little piece of the virus's DNA and saves it, storing it in its own genetic code. This collection of viral snippets is called CRISPR. It's basically a most wanted list, and if that virus ever comes back, the bacterium uses that list to guide a special protein called Cas9, like a guided missile, to find and just chop up that virus. It's brilliant. And this was the big eureka moment. Doudna and Charpentier realized, whoa, this isn't just some bacterial defense system. They'd stumbled upon a tool, a programmable tool. If they could just feed this system a new most wanted poster, any DNA sequence they wanted, they could guide that Cas9 protein scissor to cut any DNA strand, including ours, at a super precise spot. It was literally like a pair of molecular scissors with a built-in GPS. The second they published what they found back in 2012, it was like a starting gun went off. It kicked off one of the most intense, high-stakes scientific races in modern history. Suddenly, the question wasn't if CRISPR worked. The question was who could get it to work in human cells first. It was this mad dash for prestige, for Nobel Prizes, and yeah, for patents that could be worth billions. Kind of like the space race in the 50s. You had labs at UC Berkeley, led by Doudna, and the Broad Institute, led by a brilliant young scientist named Feng Zhang, just racing to be the first. And sure enough, just a few months after Doudna's paper, Zhang showed it could work in human cells. And that's when the patent war began, a battle that would go on for years. But as this amazing new technology spread like wildfire, Jennifer Doudna, one of the people who created it, was just as terrified as she was excited. I mean, she really understood the incredible power she had helped let loose on the world. This quote from Doudna herself, it's just haunting. It shows you the enormous ethical weight she felt. She had a nightmare that Adolf Hitler was asking her about CRISPR, and he was taking notes. She said she woke up in a cold sweat, just horrified, realizing that the exact same tool that could cure diseases could also be used to achieve the horrifying dream of eugenics, of creating designer babies and splitting society into the genetically rich and the genetically poor. So, the really crucial question became, where do we draw the line?
And to get into that, you have to know the difference between two kinds of editing. First, there's somatic editing. This is where you fix cells in one person, say, in their liver or their blood. The key thing is, those changes are not passed on to their children. Think of it like a tattoo. It only affects one person. But then you have germline editing, and this is a whole different ballgame. This changes the DNA in sperm, eggs, or embryos. These changes are permanent. They will be inherited by every single future generation. This is not a tattoo. This is rewriting a chapter in the family history book. Forever.
And that line, the one between the tattoo and the history book, that's where the huge debate is raging. Because this technology, it holds the potential for both incredible, unbelievable miracles and some really unprecedented dangers. So let's start with the promise, because the power for good here is just immense. I mean, take Victoria Gray. She suffered from the agonizing pain of sickle cell anemia, and she became one of the first people ever to be treated with a CRISPR therapy. Today, she is basically cured. We're seeing therapies that turn a patient's own immune cells into super soldiers that go out and fight cancer. And it's not just medicine. It's being used to make mushrooms that don't turn brown, which cuts down on food waste. They've even used it to develop super fast, cheap diagnostic tests for viruses like COVID-19. It's incredible. But then you have the dangers. And they are very real. In 2018, while all the top scientists were still debating the ethics, a scientist in China named He Jiankui absolutely shocked the world. He just went and did it. He crossed the line and created the world's first germline-edited babies. The genie was officially out of the bottle. And beyond rogue scientists, there's the risk of off-target effects. You try to fix one genetic typo, but you accidentally create 10 new ones. And maybe the biggest social fear of all, will this create a biological caste system where the rich can afford to edit out diseases and even enhance their kids, leaving everybody else behind? And all of this, it brings us to the central message of the Codebreaker. See, this isn't just a science story anymore. It's a story about all of us, and about the future that we decide to build together. Let's just be really clear about something. The technology is here. It's real. And it is not going back in the bottle. So the question is no longer if we can edit our own evolution. The question now is, how will we choose to do it? What are the rules? What's our moral code going to be? And look, these decisions are way too important to be left to just scientists or politicians or big companies. This has to be a conversation for all of us. It needs to happen in every community, in every family. We all have a stake in this, so we all need to understand the science, we need to debate the ethics, and we need to decide, together, what kind of future we really want. The book leaves us with this really powerful, really heavy thought. Decades from now, when this technology is just normal, your descendants might look back at our time, right now, and ask you, when your generation was given the power to rewrite the code of life, to literally direct the future of our species, what choices did you make? The code to write that future, it's in our hands now.